Dr. Danti is Unit Coordinator of the Italian Consortium on Material Science and Technology and Assistant Professor in Material Science and Engineering at the University of Pisa. She holds a PhD in Health Technologies, being a Scholar Research Associate at the Department of Bioengineering at the Rice University in Houston, Texas. Her research topics are biomaterials, nanomaterials, and tissue engineering. She has 14 years of experience on scaffold fabrication and biological interaction with primary human cells in orthopedics, otorhinolaryngology, and oncology. So she will present biological data on chitin. So please, Serena, the floor is yours.
Thank you very much. Good morning, everybody, and I'm very happy to be here and to show you our last results related to the biological effect on nanochitin on the supports that were given in ECOFUNCO project. So I'm going to talk and briefly give you an overview about the biological data that we have collected until now in the project. You know, chitin is a very abundant source that is entitled with many activities and can be reused and can be also can provide very important inputs to our body, to our skin, and but we have to distinguish between the chitin that we can find as a source and the nanochitin, because when chitin is in the fibrillated form on the nanoscale, can have very different features that may have different biological outcomes when we are exposed to nanochitin instead of chitins. In fact, there are many works that have pointed out that on the nanoscale, the chitin can lose its pro-inflammatory activity, and also is important to stress that can use the allergenic character. So in addition, can be metabolized by our body enzymes. We have chitinases, and lysosome is another enzyme which is involved in the metabolism of chitin. So we can also eat chitin, of course, and we can implant even inside the, if it goes inside the body, there is no problem. We can we can degrade the chitin. Then we can have a good interaction with different cells, and in particular, if we control the crystalline structure and the purity of the chitin nanofibrils, we can even improve an antibacterial activity, anti-inflammatory activity, and also we may have cicatrizing features and also anti-aging activity. So chitin in the form of nanochitin has become very interesting in the cosmetic and biomedical field. Also, chitin is not toxic, so we can use, and it's safe. It can be biodegraded in the environment, but also in our body, and also we can exploit the antibacterial potency and the immunogenic activity of nanochitin. So we have a lot of reasons to apply chitin, and to what is still ongoing is understanding very well the effect that we can have when we expose chitin to different tissues in our body, and also the different source of chitin. Another important feature is to consider when we are developing so devices or other products that contain nanochitins, is the process itself, how the process can affect the final biological activity of chitin. Okay, what I wanted to show in the previous slides is that we use electrospray, and this was explained in the meetings of ECOFUNCO by Dr. Barazzini, who was devoted to this task. And electrospray is a method to generate a spray under an electric field and to have very fine droplets that are diffused, that are traveling from the needle to the collector by effect of the electric field. By this way, we can have a fine distribution of these droplets, and if these droplets contain, for example, the nanochitin, this is a way in which we can have a dispersion of nanochitin on a surface. So we tested the different sources that were provided in the project ECOFUNCO, and of course, we have these sources provided by Cellabor into different forms. So we have the shrimp-based nanochitin, and we have the mushroom-based nanochitin. These are two different sources. One is from animal sources, and the other is from, let's say, vegetable sources. So Bahare already explained that we had to try to use different solvent systems in order to have the spray of the chitin. So of course, in this project, we aim to be eco-compatible, we aim to be ecologically friendly. So the natural solvent to use is water. So anyway, because the chitin contains is an organic compound, and also we need to generate an efficient interaction with the electric field in order to have a very well dispersed and diffused jet of droplets. We combine water with other solvents that are provided with an hydrophilic but also an organic part. And in particular, we decided to use acetic acid that still is a natural acid that we can have, and also we try another solvent that is as a fluoroisopropanol. So we try with the different solvents, we try to electrospray the chitin. I am saying this way because the solvent have finally an impact on the biological data that I will show you later. And here you see that we try to use the same conditions like concentration, voltage, flow rate, and also the same geometrical parameters for all the tests that we did, except for the solvents that, and of course for the chitin sources. And here you can see that we started to optimize first on aluminum foil, and then we used the paper tissue that was provided by Lucenze. Of course, when we put something in between the electric field, it can affect the way the chitin is dispersed on the surface. So that is why we had to provide double optimization in order to have the final product. When we check the results, of course, water was not giving the best one because we had a big droplet that were concentrated of chitin nanofibrils. Whereas with acetic acid, we could have a more finely dispersed distribution of the nanochitins, and of course, with the fluoroisopropanol, we had a good dispersion. We replicated on the paper tissue, and we observed that water was the same, so was not efficient to this process. Acetic acid was kind of good, and as a fluoroisopropanol give rise to this very finely dispersed nanofiber web. And this is because of a self-assembly of chitin on the nanoscale that is separated by the solvent and then reassembled during the process of evaporation.
Excuse me, Serena, you have 5 minutes left.
Okay.
Thank
so
you.
we'll try to be fast. So here you see the SEM, the results, and then we go finally to the cytotoxicity test that you can see here in the slide. We perform cytotoxicity test, direct and an indirect cytotoxicity test. So the first was using both are using human dermal keratinocytes that were put in touch, in the first case, with the tissue that was covered with different chitins, and in the second, they were just in contact with some supernatant that was produced by the tissue covered with the chitins. These are tests that are in the ISO that is related to cytotoxicity, and we perform. So we could observe that the cells are very viable. They could even adhere to the surface, so we didn't detect any important feature of cytotoxicity. That was also confirmed by the metabolic activity test, the Alamar Blue, that was performed on the tissues, and also we could observe under the scanning electron microscopy that the cells were able to cover the surface of the tissue that were pretreated with the different chitins. So the most important slide that I want to show you today is related to the immunomodulation of that we could detect related to nanochitins. So we could see that so we had a slightly different behavior according to the source of nanochitins that were used. In particular, the shrimp-based nanochitins, they had a higher early pro-inflammatory activity that we can see at 6 hours, for example, in figure A and B. So we see that the rise of all the pro-inflammatory cytokines, but these cytokines are later on are reduced with respect to the basal level. So we had like an initial effect that is later on compensated and becomes anti-inflammatory in the longer term. Whereas the mushroom-based chitin nanofibrils showed a weaker pro-inflammatory behavior, and they had a predominantly anti-inflammatory activity. In fact, the TGF-beta, the yellow, that is a powerful cytokine that is involved in anti-inflammatory activity, we can see that is always higher in figures C and D with respect to the other one. So this is in our considerations. This is due to the different sources that can lead to a different recognition by our cells, by human cells, if it's natural or if it's vegetable or if it's animal origin. So we can also see that there are slight difference, but overall the behavior is to be considered anti-inflammatory, and is more anti-inflammatory, let's say, at earlier times when we use mushroom. So this is an important indication if, for example, the tissue will be in touch with our skin for a short term, maybe it is more convenient to use the mushroom-based chitin nanofibrils. So we can conclude that electrospray can give rise to, if we tune the solvent systems, but also we can find natural-based solvent systems like the acetic acid water combination. We can have a good dispersion of nanochitins, and also we can have a very nice biological results in terms of cell viability, and of course we can have immunomodulation that is driven by the nanochitin, and in particular the source may seems to have a role to affect even very finally, it's not a dramatic effect, but seems to have a more pronounced anti-inflammatory activity than the shrimp base. So I finish, and thank you very much for your attention.
Thanks, Serena, for your presentation. And this was a very interesting insight on the research and application of chitin in ECOFUNCO. So thank you so much for participating today.